And get started.
Please do, thank you.
For those of you I didn't get a chance to meet last night, my name is Roy Hamilton. I'm a behavioral neurologist here. I'm also the director for the Laboratory for Cognition and Neurostimulation. And what we're going to do for the next half hour or so is talk about a couple of the principal tools that we employ in the laboratory for neuromodulation, sort of non-invasive neuromodulation. I'll also deviate from time to time, in addition to just talking about methodology. I'll also talk a little bit about some things that I think are exciting that we're working on, and then also some things that I think are problematic with some of the tools and technologies, right? And also what I think, ways I think we ought to address some of those problems in the future. Okay, so first disclosure, I am not very entrepreneurial, it turns out. I bet I have family members who wish I were, but I don't have anything to disclose related to any of the technologies that we're going to talk about today. All right, these are the tools that we're going to talk about today. Right? These are the two workhorses of the laboratory, and if you're looking through the literature on non-invasive brain stimulation, these are the two tools that you are most likely to encounter. Right, the first of these that we're going to talk about is transcranial magnetic stimulation. The second one is transcranial direct current stimulation. And although they bear a lot of resemblance just in sort of the initials and how they sound, it turns out that there's a lot that differs between them in how they are practically implemented. So let's just take a few minutes to talk about each of the methodologies in order. So let's first talk about transcranial magnetic stimulation. So TMS is based on the Faraday principle, the principle of electromagnetic induction. So reach back in the deep recesses of your memory to your college or maybe your high school physics class in which you talked about electromagnetic induction, that if you were to have, say, a coil of wire and you ran current through it, right, that for that brief period of time that you were running current through that coil, it would become an electromagnet. It would generate a magnetic field. Okay, so if you had something else, a second body that was capable of conducting electricity, and you had it very close to that thing, right, very close to that coil, as you passed current through that coil, as that coil went from being inert to having a full magnetic field, whatever that magnetic field strength was going to be being inert again, in that moment, as the magnetic field strength was fluxing, if you were to measure from that second conductive body, you would be able to measure that current is being generated, it's being induced in that second conductive body. Okay? That's electromagnetic induction. They don't have to be touching in order for the one conductive body that's having current pass through it to generate current in the other. Now imagine that the coils that we use in TMS research, that's that first coil of wire, but that second conductive body that we were talking about, that is the neurons of your brain, which of course operate via electrochemical principles. And so it is possible to generate enough current in neurons, in brain cells, to cause them to acutely fire, to depolarize. Right? And that is what transcranial magnetic stimulation does. And so it induces this current in the cortex. It's obviously non-invasive, so the point is these things don't have to touch, right, as I said, so you can administer stimulation from the outside. You don't actually have to be in contact with the cortex, and that's what allows its non-invasive property. And so we can target different areas of the brain by moving the coil around. And that's how we achieve focality with TMS. And so the size, the shape of the coil, and the fact that we can reposition it over parts of the brain that we want to manipulate. And so here's a sort of cartoonish picture of it being done. We don't always hold subjects in place as fastidiously as this cartoon demonstrates. It seems like they're sort of held in this bracket. That's not always the case, but we do try to keep subjects still as we apply transcranial magnetic stimulation. Another property of contemporary units that I'm showing here is the use of frameless stereotaxy, a neuronavigation system. So here we have an example of an infrared camera that's set up, and we have one that looks just like this in our laboratory. Many laboratories do. There are trackers that are placed on the head of the subject, the coil itself, and you use software in which you input the MRI data of the subject, usually acquired previously, right? And, the camera recognizes where the head of the subject is, it recognizes where the coil tool is because you put trackers on both of those, and it knows the MRI data, so by kind of transitive property, it kind of knows the relationship between the coil and the head, right, and places it and it situates it in the context of the MRI to show you relative to the subject's brain where you're likely to be stimulated. Right? And so now we've got this technology down to the level of millimeters in terms of precision, and which is pretty good because when we're talking about TMS, most common systems, you're talking about stimulation that has a resolution of about, let's say, a centimeter to a centimeter and a half by a centimeter and a half on the cortical surface. So imagine if you all hold your thumbnails out and look at them and imagine an effective area about that size over your cortex. Right? That's about the area that we imagine stimulating. So having millimeter precision with how you target that is pretty good. So let's talk a little bit about what some of the advantages are of being able to manipulate the brain in such a way. So coming from the background of neurology that I'm coming from, one of the key ways that neurologists and cognitive neuroscientists have made inferences about structure-function relationships in the brain is by observing what happens behaviorally in individuals who have injury to those regions, right? I mean, historically, that's how we came up with some of our sort of landmark insights about language, about different aspects of cognition, executive function. You picked patients who had deficits in those areas, there were lesions in those areas, patient HM in memory, for example. Well, it turns out that there are a bunch of problems when that is your principal way of exploring a perturbation of the brain. All right? So one problem is that brains that have a focal problem, right, often have other problems. If you have a stroke, for example, and that stroke's in a particular area of your brain, turns out that there are a number of reasons why you might have disease in other parts of your brain that are not of specific interest to the cognitive neuroscientist who is doing that study. Right? So if you were able to pick a specific spot and say, I only want to affect that spot to the best of my ability, you avoid that confound. Another problem is that brains, when you do a lesion study, when you are studying someone who has a hole in their brain and it causes them to behave differently, you could think about it as a study of not specifically what that area of the brain does. It's a study of what the rest of the brain does when it's missing that one area. And it turns out that one of the things that the rest of the brain does when it's missing a brain area is it changes. It changes how it functions. Right? And maybe you're not specifically interested in that change, you're interested in that region. Well, in that case, you would want to study whatever the changes are or whatever happens at the moment that you have that lesion, at the moment at which that area was injured, rather than months out when the brain has had a chance to adapt to it. And so TMS affords you that too, right? It allows you to acutely study the brain after you've perturbed a region before it has had a chance to adapt or sort of develop other strategies and other sort of computational routes to accomplishing tasks. It also affords you a variety of methodological sort of conveniences when performing study. For example, it allows you to repeat the same study in the same subject in an on state and an off state. Imagine the lesion study, contrast this to the lesion study where it is impossible. The genie is out of the bottle when you meet the patient, right? You can study them after they've had their lesion, but at that time, it is impossible to study them before they've had their lesion. And then also, it's possible to study multiple subjects with the same manipulation and the same essential lesion, the same perturbation. One difficulty in lesion studies is that while you can find groups of subjects who have damage in the same area, subject A may have damage in an area and the area adjacent to it to the anterior, subject B damage to that area and an area adjacent to the posterior, right? It's hard to find subjects who have exactly the same perturbation, right? And so this way it allows you to standardize across multiple subjects. So while we don't often use this language of virtual lesion because it implies that TMS actually causes permanent disruption, which it certainly does not, I do still sometimes think that as a way to cause these focal disruptions in a way that's more convenient than the most historically tried and true way that neurologists and neuroscientists have studied focal perturbation. So now I want to talk about the use of TMS and how it fits in the context of other neuroscientific tools in cognitive neuroscience. And here I'm going to focus on a couple of aspects. What I have to say here I think applies to TDCS2. So I'm going to use this graph, which is, I think it's aging well. It's 2000, so a number of years ago now. But what it shows on the x-axis is the spatial resolution of a technology in cognitive neuroscience. And it can range, right? It can range from, and I realize maybe you might not be able to read this all the way in the back, and unfortunately the screens are blinking a little bit, so it might be challenging. So I'm going to read you some of these, right? So your neurophysiologic technologies like EEG, ERP, and MEG, their spatial resolution is not so great compared to, say, on the other end of the spectrum, things like single cell recordings, right? On the y end of the y-axis, you've got the temporal resolution, where here you're ranging from milliseconds to years. And here your neurophysiologic tools have a lot of power, right? Your EEG, ERP, they're measuring things down to the millisecond, right? Whereas other studies are measuring things over longer spans of time. So for example, conventional imaging studies, like a CAT scan of the brain, is not a high temporal resolution technology. It's not going to show you differences from moment to moment. So you could situate TMS somewhere in a relatively sort of moderate spatial resolution frame, and because you can control when you administer TMS and measure its results both physiologically and behaviorally, it has pretty good temporal resolution. But the key thing that I want to point out, and this is true of both TMS and TDCS, is a third axis, one that's coming in and out of the board, if you can see the sort of three-dimensionality of the graph. And what that represents is the difference between correlation, which many of our cognitive neuroscience technologies feature. So a subject performs some tasks, they behave in a certain way, something happens in the brain, you measure what's happening in the brain contemporaneously, and then you associate the thing that happened in the brain with whatever behavior was going on, and you try to make inferences on that basis. That's a correlation. Versus interference. What I mean by that is some person is doing something, you actively do something to their brain, and then you measure what that does to the behavior. Right? It's a different direction of inference. And it's a very powerful one because you cause change in the brain and see how that causes change in the behavior. So that allows for different kinds of inferences, and I also think is very complementary with some of these correlational tools. Imagine figuring out an association between some behavior and some pattern of activity in the brain, and then pairing that with the discovery that if you then perturb that activity of that area of the brain using one of these technologies, you can disrupt or somehow change those behaviors, right? That's a very powerful combination. All right, so there are a variety of techniques used in TMS to explore cognition, just some basic methodology. I'll run you through a handful of some of these in case you're reading TMS papers. The oldest way to administer TMS in the context of behavioral tasks is single pulse TMS. That is, someone is doing a task, and in the midst of doing the task, as each trial as they're doing it, at exactly the right moment, pow, you administer your TMS to the spot in the brain that you think is somehow relevant for the behavioral task that they're performing. Okay. Now, this has a certain disadvantage, which explains why it was more used early on than now, which is that's great, but you kind of have to know when in the process that area of brain that you're interested in is implicated, right? When is it most relevant for the task at hand? Because if you're only applying a single pulse of TMS, you're going to have a pretty narrow window, tens of milliseconds, in which you're causing a very transient disruption. Now, you can do a little bit of methodologic judo and turn that disadvantage into a kind of way of doing a TMS study if you want. So let's say, for example, you've got a very strong theory that a particular part of the brain is engaged in a particular cognitive task, but you don't actually know when in the process it is. One thing you could do is something called a chronology study or chronometry study. You could administer single pulses of TMS at different times relative to the presentation of the task. And so then you could demonstrate across the lifespan of the task that, oh, if I administer it too early, I don't get an effect. If I administer it too late, I don't get an effect. But if I administer it in this Goldilocks zone, that's when my TMS seems to have an effect. So that's when I think this is, it's relevant in terms of the time span of the task, right? So you could use that weakness as a strength. You'll more commonly see nowadays studies that feature repetitive TMS or RTMS. So an RTMS it's the presentation of patterned stimuli at a particular frequency. And there is a received wisdom here where low frequency TMS, often in the range of 1 hertz, so 1 pulse per second, tends to be associated with inhibition of cortical activity, cortical function. And the way this is usually used, it's usually used as in the context of an offline study. So you're administering TMS for some period of time. Let's say 20 minutes of 1 hertz TMS. The person's just sitting there getting pulses at a very low rate. And then that results in some residual after effect, often inhibitory in nature, during which time you're having a person either undergo whatever physiologic measures you're interested in or some behavioral task. You can also administer high-frequency TMS, usually in the neighborhood of above or around or above 5 hertz. And here, it's felt that this is facilitative with respect to cortical activity. Now, I want to be specific and say cortical activity, right, as opposed to behavior. It isn't always a one-to-one correspondence between brain areas being more active and you being better at some task. Right? Brain is a finely balanced thing, so it's not always that more is better with respect to brain activity and less is worse. And then you'll also see in the literature brief trains of TMS. And brief trains, for all intents and purposes, you can think of them as being like single pulses of TMS, but more of them. Okay, and so you get around that you get around that problem of having a very short window of activity. You expand that window by giving a train of TMS pulses in the context of trials of a behavioral task. Right? So it's a little bit more robust in that way. A technique that has really asserted itself in the field in the last several years, something called theta burst stimulation. And what theta burst stimulation is it is bursts of TMS. So if you go back and you start reading papers, this is what they're talking about. So it is triplets of TMS, so 3 pulses of TMS administered at very high frequency, 50 hertz. Right? So if you were running them back to back, there'd be 50 of them in a second, pretty fast. But what you're doing is you're going to give 5 of these little bursts per second. So it's 3 bursts at 50 hertz, but 5 of them per second, that's 5 hertz. So you give bursts of bursts. And it turns out that, at least in the hands of some people who were using it, it has fairly robust effects that can be either inhibitory or facilitative with respect to cortical physiology and activity. And so the one other key advantage of it is that you don't have to administer it for anywhere near as long a period of time as some of these other TMS techniques. So a couple of minutes compared to, say, 15, 20 minutes for some of these other techniques. And that's, I have to say, is probably why it's become so popular of late. And I don't have time to actually go into all the ways in which TMS is now being combined with other technologies. Just take me at my word that recently TMS has been playing very well with others. It used to be the case that it was thought to be methodologically impossible to simultaneously stimulate the brain with a magnet with sort of overwhelming amounts of, super threshold amounts of current, and also measure the tiny amounts of current that the brain generates in order to measure EEG. But we cracked that nut. And then also, it was previously thought to be an engineering impossibility to take a magnet, stick it in another magnet, like MRI, and discharge it while you're acquiring MRI data. That problem has now also been solved, so there's fMRI compatible TMS. So it can be applied in the context of acquiring some of this correlative data that we were talking about, right? So we can now pair those two halves, pair those two ways of making inferences about brain function. So I want to say just a quick word about one thing that I think is kind of exciting in brain stimulation. It's going to segue into your next talk, which is by my colleague in the TMS lab and network neuroimager, John Medaglia, and it's going to touch on work that he and I have been doing. A lot of it him, but I think it's exciting because it sort of it points to how advances in how we think about brain organization influence how we think about neuromodulation. Because I think those two things are so yoked that you can't really talk about what the advances are in one without thinking about the advances in the other. Right? So what I'm showing here, and I don't want you to get caught up at all in the actual data of what I'm showing. All right? I'm just being illustrative. What I'm showing here is how we approach some of the work that my lab does primarily, which is in the language domain. I'm very interested in language function. And so we've been stimulating individuals, in this case stimulating people with strokes and aphasia, people who've lost their language ability, and demonstrating that we can, that they have multiple areas in their brain that seem to be active when they're performing language tasks that healthy individuals don't activate. But B, if we target one of those areas, we can change their ability to name. But we're left with an open-ended question, which is, there were a bunch of areas, but only one of them seemed to work. Right? So why is it that stimulating one area worked and stimulating these other bright blobs on fMRI doesn't seem to work? What are the principles that should inform us as to where we ought to go with brain stimulation? And so the critical insight here is that it should have something to do with differences between how these areas are hooked up, how they're connected. Right? That it's should be an artifact of, or a downstream result of the brain's connectivity. And so we've been thinking a lot about that. And again, I don't want you to get too hung up on this specific example, but in the language domain, we've been thinking a lot about how language is controlled and thinking about it in terms of network properties of the brain. Right? In terms of, well, if you've got a particular site in the brain, again, sort of reducing things to a kind of cartoon that will be gone over in more detail, I'm sure, in a half hour. You might imagine an area that has a certain kind of network property as functioning very different than an area that has this kind of connectivity than an area that has a third kind of connectivity. And again, I'll gloss over the details, but just looking at these kinds of examples, you can imagine how they would have different roles. And so what we've been interested in doing is figuring out whether we can determine, based on theories about how these networks, their structural organization and connectivity should dictate their roles, right? If we can use that information to determine who we ought to stimulate and, importantly, where we ought to stimulate them for particular properties that we want to modulate. So, for example, this is, again, alluding to work that we've been able to do in the lab. This is sort of work that led up to it. Dr. Medaglia's been involved in this kind of work too, but it's showing different maps that look at different kinds of properties of network control and showing that, well, okay, an area that has these kinds of properties of this node, we'll sort of give it this label, it's that it maps out that there are different areas of the brain that have those properties compared to an area that might be a little bit more reminiscent of how this area is connected, that might have sort of more specific network connections, versus an area that sort of joins large modules, and that the maps of brain areas that exhibit those properties may differ, not just between individuals, but not just within brains, within individuals, but also between individuals at the same site. So just to, and so that gives us some hypotheses about language control, how then the structure of the network might define that control, and how also, after injury, that these properties might tell us something about where we ought to be stimulating. And so just to give you an example, in healthy individuals, we started looking at particular tasks that I'm going to very sort of broadly refer to as open and closed, where the tasks that are sort of closed in their nature, you give a prompt, And the answer has to be very specific to the prompt. So for example, if I give the word car, there are relatively few things one does with a car, and I ask you to come up with a verb for what you do with the noun car. You're heavily biased for coming up with the word drive, right? So we thought, well, maybe networks, areas in the brain that are more inclined to this kind of controllability that's more specific, maybe they have that kind of property too, that stimulating those areas will do something when the task demands are very specific. But maybe when the task demands are very open, what do you do with a ball? Well, you hit it, you throw it, you kick it, you can probably think of 5 other different things, right? But maybe that requires a different kind of node. And so we, in the interest of time, because I know I'm running a little behind, we defined these networks, right? And we're able to generate maps that on an individual basis showed how well they characterized these particular network properties. And what we could demonstrate was, indeed, that when a language task was open in nature, that it had these open task demands, that if the area that we stimulated was high in this property of boundary control, right, the kind of area that we hypothesized would be required for these kinds of open tasks, well then zapping that area perturbed your performance on that task. If the same area, by contrast, was high in this other kind of control, this was for more specific control, well then in the same physical area in other individuals, right, you would get control, you would perturb the more specific task. The point is, in the same area of the brain, depending on how it's hooked up, depending on its connectivity from individual to individual, we can start to make predictions of how the behavior will be changed. Right? Not just observations, predictions of how your behavior ought to be changed by TMS. So anyway, that's just a detour and something I think is particularly cool about where we're going with this. Let's switch to the other technology that we use in this laboratory. Transcranial direct current stimulation, the way it differs from TMS, it's sub-threshold in its nature. Remember zapping the brain so that the brain cells actually fired? This actually administers current at such a small amount that it just nudges brains a little closer or farther away from their firing potential. Right, so what the net effect of that is to alter their firing rates. It can be administered in different ways, and the received wisdom is that the polarity of the electrode that you use over their brain is going to give you different effects. And it has a bunch of practical advantages, right? It's cheap, it's portable, it's easy to pair with other therapies. And it's very safe. So, don't have time to explain this cartoon. What it shows is that if you put the electrode over the brain, right, what you're doing is you're inducing charge separation. So different parts of the neuron are different distances away from that electrode. So some parts will be driven to be more positive and some parts will be driven to be more negative. And because that has a differential effect on, different parts of the neuron have different effects on how effective they are in inducing the neuron to fire, that sort of biases them one way or another towards or away from firing. It's thought to have several mechanisms of action, including modifications of synaptic microenvironments by things like long-term potentiation. It also is affected in complex ways by other neurotransmitters. So medications that you might be taking, they're psychoactive, can affect how DCS works. Obviously, if you're stimulating one area of the brain, because of the connectivity of the brain, you're going to affect other areas of the brain. So connectivity-driven remote effects might be in play. And remember that your brains aren't just neurons. There's lots of other stuff in there, and running a bunch of current in it might affect other things. Okay, this, as opposed, remember how much time I spent talking about how focally you can stimulate with TMS? This is the opposite of that. Right? If you apply the electrodes to the scalp in some of the most common montages, you run current between you, this whole smear of current that runs through much of the brain. It's one of the limitations of the technology. And where you place the electrodes matters a lot. So it's typical to place one over an area that you're interested in, but then people sort of go willy-nilly with where they place the other. They can refer to it as a reference electrode, but it turns out that where you place that reference determines the direction that the current's going to flow. There are newer versions that employ what's called high-definition TDCS. They achieve some greater focality, but it's still very limited compared to TMS. And people have been exploring it for a variety of things. I think in part because of the low barrier to use, right? It's easy to use, cheap, and portable. And so people are exploring it for practically everything. In our laboratory, we've explored for a variety of different things. I don't actually have time to talk to you about all of them, but a couple of them include creativity. For example, the ability to come up with novel uses for common objects. That's work that we did with Sharon Thompson-Schill here. Some work that we're still doing and have been working on with Adrian Rain is the ability of TDCS of the frontal lobe to dorsolateral prefrontal cortex and other areas of to mediate or to modulate violent intent, the intent to commit violent acts. And so this technology has really taken off, right? That's just a graph of how many papers there have been over the years. So much so that unlike TMS, it's now actually being marketed straight to the public. So this is a company, Martha showed a slide of it yesterday, that is marketing to people who want to improve their athletic ability, in part by stimulating either this particular group, frontal areas. There's another group called Halo that stimulates motor areas of the brain. There's also a do-it-yourself stimulation community. Again, the technology is so cheap, has such a low barrier of entry, there are people out there who are doing it to themselves, which, as you might imagine, might be of ethical interest, social interest to your group. Now, for TDCS, there is some cause, I have to say, for skepticism. This is something that's emerged over the last couple of years. Papers that suggest that maybe the stimulation doesn't have as reliable or reproducible an effect as we would like. And we actually got into a sort of back and forth with some of these authors about whether it does or doesn't. But that's not the point. The point is, if enough of these come out, it gives you pause as to whether or not there are problems with the reliability of this technology. And when members of our own group came out with sort of negative meta-analyses as well, sort of raised these questions even further in our own minds. So I think a lot of it, though, has to do with the fact that there are so many degrees of freedom with this technology. There's so many things you can vary, and we don't understand all the implications of all of them, or any of them, that it would be surprising if they had robust and completely consistent effects. So, amongst the things that vary a lot that we still don't understand very well: location, size of electrodes we use, the shape of the electrodes we use, the intensity we use. Here's a graph that shows that if you apply the same stimulation at two different intensities, you get opposite direction effects. So, the effect on the brain is not linear, right? Something else is going on. The actual effect of polarity on the brain, the effect of duration on the brain, the effect of the period between sessions and the total number of sessions, all things we don't yet fully understand. I'm particularly interested in the effect of what you are thinking, like the actual state of cognition during stimulation and how that influences stimulation. So in this particular experiment, we varied whether or not subjects were performing an easy or a hard working memory task while we're stimulating them and assess their performance on a different working memory task after. And what we demonstrated was the same task. We just modulated the task difficulty. We could get very different effects, right? What you're doing at the time of stimulation really matters. The thought does count. So those are known unknowns, and then there are a bunch of unknown unknowns that we have to ferret out. So the unintended effects of cognition on other cognitive domains. It tends to be the case in cognitive neuroscience studies that we do a study where we explore one thing, but we're giving broad stimulation. So what happens to everything else, right? And then the interactions of other kinds of things like drugs and other technologies, that we have to explore. So where I think we are right now is that TDCS is in the middle of a ride on the Gartner Curve. Okay? So some of you are familiar with the Gartner Curve. When you introduce technology to society, there's this immediate period of inflated expectations. Like, this technology can do anything. It can help me play video games. Let's market it to the public. Right? And then after that, when they discover that it doesn't do everything, there's this reaction formation. TDCS can't do anything. Right? And this sort of wave of skepticism that washes over the public. But I think there's too much data right now to suggest that it does nothing, and there's certainly too much data to suggest it does everything. And so I think we're starting to enter a plateau of productivity, that TDCS does something, and it's up to us in the future to figure out what that thing is. So, TMS and TDCS, vital tools in cognitive neuroscience and cognitive neurology, for the reasons that we articulated. Evidence suggests that both methodologies can facilitate neuroplastic changes in brain activity. I talked a little bit about what I was interested in the future that I think is exciting, like bridges to the next talk, network properties. And then for TDCS in particular, I think that poorly characterized stimulation parameters are important and addressable sources of variability in non-invasive brain stimulation. All right. Woo-hoo!